Good afternoon, everyone. Today's lecture topic is cancer drugs. And before we start discussing the drugs, let's talk about the cancer and cancer development. And most of the time, the cancer is developing from one single cell that starts division frequently, and most of the cancer drugs are directed to fast dividing cells. Unfortunately, there are also other cells that divide fast, like for example epithelium, and uh, that is why there are many adverse effects and also hair. So those tissues that grow continuously throughout the life. So that is why there are so many adverse effects with the cancer drugs. And most of the drugs are directed to the cell division, and that is why let's talk about the cell cycle before we start talking about the drugs, because the drugs are divided to two categories. Like first category is cell cycle specific drugs, and second is cell cycle non-specific drugs. The cell cycle specific drugs will influence one of the cell cycle stages, division stages, and will only effective if the cell cycle is in that specific phase. Cell cycle non-specific drugs will be effective uh, non-dependent of the cell cycle phase. So for the any cell, there are um, there are uh, several phases, and we start from the phase G1. During this phase, a cell will prepare proteins and enzymes required for the cell division and also for the DNA replication. In this stage, cell can usually also increases in size, and after G1 it can either go to G0, that is a dormant cycle, so the cell will stop division for a while, or go to the S phase, that during which the replication of the chromosomes, uh, the, uh, the replication of DNA occurs, and the cell will produce a copy of the chromosomes. So after the S phase, the cell go to the G2 phase. During this phase, the chromosomes will be prepared for the next stage, that is M phase, or mitosis. Mitosis will complete the cell division and is further divided to the uh, four phases, that is prophase, during which uh, the nuclear envelope is removed and the chromosomes will freely float in the cytoplasm of the cell, and then metaphase, during which the mitotic spindle is produced and connected to the chromosomes, and chromosomes will be aligned in the center of the cell, and then anaphase, during which the chromosomes will be uh, separated by the mitotic spindle to the opposite poles of the cell, and telophase, during which the cell is completely divided and mitotic spindle is removed. So after completion of these phases, the cytokinesis occurs, and new cell can either continue this cycle or become dormant. So this is the cell cycle. I'm sure you studied that on biochemistry, but we just revised the knowledge. So and to ensure that every cell that is produced during this cell cycle is identical copy of the previous cell, mother cell, so, there are 3 checkpoints that will ensure that everything goes okay, there are no any abnormalities or some um, or mutation occurs or something bad happen with the cell. So, and main checkpoints are at G1, G2, and M phase. So these are correspondingly called G1, G2, and M checkpoints. And if we talk about the drugs, there are drugs that influence the different parts of the cell cycle, and let's start with the G1. Like G1 is a process during which a cell is preparing proteins and enzymes required for uh, the cell division, and for that proteins to produce these proteins, asparagine is a vital building block. So, and cell will require much of the asparagine. So that is why if we um, uh, if we inject an enzyme asparaginase that is destroying the asparagine to aspartic acid and ammonia, that will decrease the amount of the asparagine, and that why the cell will stuck in G1 phase, because the proteins that are required will never be produced. And this asparaginase can be used for the treatment of the leukemic cells that specifically have high concentration of asparagine, and unfortunately, asparaginase may lead to the uh, allergic reaction. And for those patients having allergic to the asparaginase, PEG asparaginase, that is polyethylene glycol derivative of the asparaginase, is produced, that has longer half-life and better tolerance and will not lead to the allergic reaction, unlike asparaginase. So this is one of the first drugs that is cell-specific, cell specific drug. Then we also have anti-cancer drugs that influence the production of the hormones, and these drugs will be effective for those cancers that are hormonal or hormone dependent cancers, and this is usually breast cancer and testis cancer, and also uterus and ovaries cancer. So the production of the estrogen in women, so the most commonly hormone that will lead to the hormonal cancer is in women is estrogen, and estrogens are binding to the estrogen receptor that is located inside the cell. Then this receptor, two of these receptors activated by the estrogen, will go to the dimerization, and this dimer will enter the nucleus and affect the uh, transcription and translation of the genes. So and normally that leads to the growth and proliferation, because estrogen is a hormone that increases growth. So that is why by blocking the production of the estrogen or influence of the estrogen, we can reduce the growth of the cancer. So, and there are different ways we can do that. So first of all, we can influence by the pituitary and hypothalamus, because the production of the estrogens in that is controlled by the pituitary and hypothalamus, and the uh, hypothalamus is producing and the tropin gonadotropin releasing hormone that is stimulating the producing of the follicular stimulating hormone and luteinizing hormone in pituitary, and these hormones are directly influencing the ovaries to increase the production of the estrogen. The ovaries are the main organ of the estrogen production in women. And we can use drugs leuprolide, gazareline, and triptorelin that are gonadotropin releasing hormone agonists, and they desensitize the receptor for the gonadotropin releasing hormone, and that will lead to the decrease of the action of the gonadotropin releasing hormone and less follicular stimulating hormone and luteinizing hormone production and release, and that will decrease estrogen production. So this is one of the ways of the regulation through the hypothalamus. Then we can block the production of the estrogens, that is aromatase inhibitors, anostrozole, letrozole, and exemestane. These drugs inhibit the enzyme aromatase that produces estradiol from testosterone and estrone from androstenedione. And estrone and estradiol are the main estrogens that affect the in a woman's body. Estradiol is mostly produced in non-pregnant, while estrone is mostly produced in pregnant women. Both are produced by the ovaries. And also we can use a selective estrogen receptor modulators, that are tamoxifen and raloxifene. These drugs will bind with the estrogen receptor and prevent the dimerization and further entrance to the nucleus. And also we have a estrogen receptor down regulator, fulvestrant, that is binding to the receptor and decreasing the number of receptors. That is why that leads to the less effect of the estrogen. So these are the main groups of the drugs, anti-estrogenic drugs, that are used for the treatment of the breast cancer in women mostly, but also can be used for the treatment of the other types of the hormonal dependent cancers as well. Then in men, we also can use the same approach, but for the decrease in the testosterone production, because testosterone production is usually reason for the testis cancer in prostatic cancer in men. And prostate cancer is dependent from the testosterone synthesis, and the same we can do with the almost same with the uh, hypothalamus regulation, but instead of the sensitization of the receptors, we use antagonists of the gonadotropin releasing hormone, because if we start to stimulate the gonadotropin releasing hormone, that will increase testosterone level so much at the beginning, so that will lead to some non-reversible adverse effect. So that is why in men it's better to block the gonadotropin releasing hormone affected by the antagonist, that is degarelix, and that will decrease luteinizing hormone production and testosterone production in testes. The testosterone, further we can block the androgenic receptor by the block anti-androgens like flutamide, bicalutamide, and nilutamide. So this is same as the selective receptor estrogen modulators with the estrogens, and also we can reduce the production of the testosterone from the precursors, that is by blocking the cytochrome 17A1 enzyme by the abiraterone. So all this will lead to the decrease of the testosterone action. Okay, then adverse effects of the drugs include the loss of for the asparaginase and PEG asparaginase, loss of the appetite, and the weakness, tiredness, the painful gums, pancreatitis, and blood clotting, increase in the blood clotting. With the aromatase inhibitors and agonists and antagonists of the gonadotropin releasing hormone, we have the decrease in the mood, the tiredness, the painful muscle pain, hot flashes, and osteoporosis. And also with the selective estrogen receptor modulators. Additionally to these, for adverse effects, we have also ovarian cancer and the heart, increase to the heart, load to the heart, because of the androgen, increase in the androgen. And for the anti-androgens, in men we commonly have gynecomastia, increase in the breast size, that is unusual in men. Also hot flashes and sexual dysfunction, or like impotence, and also osteoporosis. So these are the drugs that influence the hormones and also aromatase inhibitors. Then let's talk about the drugs that influence the cell-specific, cell cycle-specific drugs that is act on the S phase. So during the S phase, the DNA replication occurs. So the DNA, identical copy of the DNA is produced with the help of the enzymes topoisomerase that will uncoil the DNA, and helicase that will divide the two strands of the DNA. Then on each strand, the complement second complement strand is produced from the free nucleotides by the DNA polymerase, another enzyme that is required. So after that, the specific sites will be merged and then called again. So this, and for this process, the key blocks that are required are purines and pyrimidines. For the purines, we have adenine and guanine, and for the pyrimidines, cytosine and thymine. So most of the drugs will affect either for the enzymes or for the analogs of these purines and pyrimidines, or pyrimidines. So let's talk about the purines synthesis influence. And purines, both adenine and guanine, are synthesized from the inositol monophosphate that is produced from the pyrophosphate protein, amino acids, and folate. Peroxidase, pyrophosphate, amino acids, and folate. So IMP is produced first, and then can be from IMP further sonazine monophosphate, guanazine monophosphate, desoxyguanazine monophosphate, and finally desoxyguanazine triphosphate is produced. And then from the inositol monophosphate we can produce adenyl succinate, and then AMP, desoxy AMP, and desoxy ATP. So these are both desoxy ATP and desoxyguanazine triphosphate are required for the synthesis of the new strand of the DNA. Then for the pyrimidines, de novo synthesis, the orotate and same peroxidase pyrophosphate is used to produce uridine monophosphate, and from UMP we produce UTP, CTP, and desoxy-CTP. And for the thymidine, we first produce desoxy UMP. Then with the help of the thymidylate synthase, we produce desoxy thymidine monophosphate. So for this reaction, folic acid is required, specifically methylene tetrahydrofolic acid that is produced from the tetrahydrofolic acid, and tetrahydrofolic acid is produced from the dihydrofolic acid by the enzyme dihydrofolate reductase. Both of these enzymes are also target for the drugs. And from desoxy TMP we produce desoxy TTP, and also that is required for the DNA. So this is de novo synthesis. Those proteins and pyrimidines can also be obtained from the food or from other tissues, but mostly they are produced from the de novo, so like from scratch. So let's look at the drugs. So those drugs that are anti-metabolized, so they will look like purines or pyrimidines. So, and here they are. For the purines, we have first of all mercaptopurine. That is name is easy to remember because it's purine also, but mercaptopurine or 6-mercaptopurine is the chemical name for the drug. The drug is prodrug and should be metabolized by the enzyme HGPRT for the TIMP, and further it will be metabolized to TGTP and T-desoxy-GTP that are both used for the production of the RNA and DNA consequently. Also, this TIMP will block the reactions of the IMP because it will be used instead of the IMP, and that that way will reduce production of the DGTP and the desoxy ATP. Then we have uh, the enzyme, hydroxyurea is a ribonucleotide reductase inhibitor that will block both production of the desoxy-AMP and desoxy-GMP. Then also fludarabine, that is also a prodrug that will convert to the fluoro-Aura ATP that will block the ribonucleotide reductase 2. And cladribine also converted to the 2-cladribine ATP, and that will block both the ribonucleotide reductase and also DNA polymerase. Also, cladribine ATP will replace the normal DATP and will lead to the will stop the production of the DNA. So also it will decrease NAT and ATP as well. That will also result in the DNA production decrease. Okay, then let's move to the pyrimidine synthesis. For the pyrimidine synthesis, we have fluorouracil that is close to the 6-mercaptopurine. 5-fluorouracil is also prodrug that is converted to the 5-fluorodesoxy-UMP and 5-fluoro-UTP. That this will have two effects also. They will block the thymidine synthase enzyme that is required to produce the desoxy-TMP, and they also will replace the UTP. So instead of the UTP, RNA will be synthesized from the 5-fluoro-UTP that will lead to the non-working RNA. Then we also have hydroxyurea that will block the ribonucleotide reductase enzyme. And then we have capecitabine. Capecitabine is a also prodrug because 5-fluorouracil is injectable only, and the bioavailability with oral availability is awful. That is why the capecitabine was produced. That is converted to the 5-fluorouracil by the enzyme thymidine phosphorylase after the absorption in the intestine. So that increases the bioavailability and also makes oral availability for the patients, and there are less adverse effects with the capecitabine and better compliance. Fortunately, the thymidine phosphorylase is enzyme that is required for the conversion is increased in the cancer cells, and that is why capecitabine concentration in the cancer cells is higher than in the normal cells. Then we also have gemcitabine that is converted to the gemcitabine FCBP, and then desoxy FCTP, and will incorporate it to the DNA. And cytarabine that is converted to the RRCTP and will block the DNA polymerase, and also will be incorporated to the DNA. And this incorporation will not stop the DNA production. That is why there are more, it's harder to fix these incorporations, and that will lead to the mask death. That's called mask death with the DNA production. And we also have a methotrexate, that is an anti-folic acid synthesis inhibitor. Methotrexate is also a prodrug that will produce a polymer with the glutamine, and it will block the thymidine synthase and dehydrofolase reductase. That is first is required for the synthesis of deoxy TMP, and second, dehydrofolate reductase required for the reduction of the dehydrofolate to the tetrahydrofolate, because the tetrahydrofolate is converted to the dehydrofolate during the reaction. Side effects of the cancer drugs include most of the common are nausea and vomiting, the gum pain, hair loss, because hair is growing and cells are dividing as fast as the cancer cells, the tiredness, and also the blood production reduce, and further complications as bleeding, and also flu-like symptoms. So that's it for today. Thank you very much for watching, and see you next week.